Well, thank you very much for the organizers for letting me share my work with you today. My name is Scott Biering, and I'm a postdoctoral scholar at University of California, Berkeley with Eva Harris. And today I'm going to share with you some of our work identifying receptors for non-structural protein 1 of dengue virus. So dengue virus is a member of the Flaviviridae family. These viruses are enveloped with a positive sense RNA genome, about 11 kb. And this family contains many medically relevant viruses, including West Nile virus, Japanese encephalitis, yellow fever virus. And they're transmitted by Aedes mosquito. And dengue is really unique because about 1 out of 2 people live in areas that are endemic for these mosquitoes and thus are at risk for infection. So the disease manifestations of dengue range from non-severe to severe, and this severe disease is characterized by endothelial dysfunction and vascular leak. So this dengue vascular leak was originally explained as just a general cytokine storm resulting from uncontrolled viral replication. And the triggers of this, besides calling it a cytokine storm, were generally unclear. But recently, our work and others have demonstrated that the dengue virus, non-structural protein 1, which is actually secreted from infected cells, plays a direct role in this pathogenesis. So NS1 is definitely one of the most interesting proteins I've ever worked with, both biochemically, multifunctionality, and just involved in pathogenesis. But it's critical for viral replication, localized in the replication complex as a dimer associated with membranes. But it's also actively secreted from infected cells, and you can see the secreted hexameric form here that contains a lipid cargo to sequester away its hydrophobic domains. Now, levels of NS1 that circulate in the blood of infected patients actually correlate with disease severity. As you can see here, patients with dengue hemorrhagic fever, a more severe form of dengue, have higher levels of NS1 compared to patients with less severe form dengue fever. Now, the work of us and others have shown many times that extracellular NS1 contributes to severe disease by functioning as a virulence factor or a viral toxin. And it does so by activation of immune cells and platelets, inhibition of complement, and through a direct mediation of barrier dysfunction in a cell-intrinsic manner. And the reason that this is important is because you can protect mice from lethal flavivirus challenge through either passive transfer of anti-NS1 antibodies or by vaccination. So this is a critical pathway. So what do we know? The work of, my work during my postdoc as well as the work of others has been studying the cell-intrinsic pathway by which NS1 circulating in the blood interacts with barrier cells like endothelial cells. So we have the endothelial glycocalyx, which is a critical component of barrier function, as well as the tight and adherent junctions here. So we know that NS1 must first bind to proteoglycans at the surface of these cells and then engage a proteinaceous receptor. This proteinaceous receptor is unknown and would be important to understand the tropism of NS1 and flavivirus pathogenesis. Followed by, after receptor engagement, NS1 is internalized into endothelial cells through clathrin-mediated endocytosis, where a couple of events occur. One is activation of enzymes that then degrade the endothelial glycocalyx, activation of kinases that disrupt the tight and adherent junctions, and a transcriptional response that enhances this entire process. This results in barrier dysfunction, by which the endothelial glycocalyx and the tight and adherent junctions are disrupted, and you have passage of fluids and solutes across this barrier. Now, the cells do not die when they experience this dysfunction. It's actually a reversible barrier dysfunction in which we then can track a transcriptional response that leads to recovery of this pathway. So as I mentioned before, the main question that I wanted to ask for this project was what are the receptors that lead to clathrin-mediated endocytosis of NS1 into these endothelial cells? So we tried many different strategies to identify these receptors, and what ended up working was actually an RNA-seq experiment where we reasoned that if this was a transient process that didn't kill cells, the receptor would be downregulated upon internalization of NS1 and then subsequently upregulated upon return to homeostasis. So in collaboration with Charles Chu and Dustin Glasner at UC San Francisco, we conducted RNA-seq of human pulmonary microvascular endothelial cells treated with dengue NS1. And one of the factors that we found upregulated that regulated, and enter today's hero, is the beta-2 adrenergic receptor. So the beta-2 adrenergic receptor is a cell surface G-protein coupled receptor that's involved in many diverse cellular pathways. This includes pathways, this is B2AR right here. This includes in G protein-dependent manner by producing second messengers like cyclic AMP through proteins like adenylyl cyclase. But then it also has a G protein-independent mechanism where it signals through beta-arrestins to activate pathways like MAP kinase and PI3 kinase. So now what I'm going to show you is a series of experiments that I hope will convince you that B2AR functions as an NS1 receptor and is important for dengue pathogenesis. So these assays will be primarily immunofluorescence-based, and we will monitor levels of endothelial glycocalyx components on the surface of endothelial cells, specifically sialic acid, as well as NS1 internalized into these endothelial cells. So the first thing I'm going to show you is an antibody blockade experiment, where in green I'm showing you internalized NS1, and you can see an antibody, an isotype control, IgG, does not block internalization of NS1 into these cells, while an anti-NS1 antibody completely abrogates this process. An antibody against a different receptor identified in our screen, nerve growth factor receptor, NGFR, does not block this process, but an antibody against B2AR abrogates this process. So here we think that B2AR is involved. Using small molecule inhibitors of this process, we can also monitor this. Here I'm showing you an EGL disruption assay. In red, we see levels of sialic acid on the cell surface. Following DV2NS1 treatment, you can see a clearance of this barrier. And when we treat these cells with isoproterenol, a B2AR agonist, which depletes B2AR from the surface of cells, we can see that NS1 is no longer able to disrupt the barrier. And when we use a series of beta blockers that are all FDA approved, any beta blocker that is specific for B2AR abrogates the capacity of NS1 to degrade this endothelial glycocalyx, but beta blockers that are specific for a closely related yet different GPCR, B1AR, are not able to do this. Genetically, we can also show this by knocking down or knocking out B2AR. Here you can see the capacity of NS1 to degrade the EGL is abrogated when we knock it down or knock it out. So genetically, we can also show that B2AR is involved in pathogenesis and also internalization in data that I'm not showing you. But then moving on to the question of sufficiency. When you're talking about receptors of, or principles of receptor biology, you don't want to just ask if it's required for internalization. You also want to ask if you can find a cell where it's absent that doesn't normally do your function. If you add that back, can we then see this, this process return? So we found a couple different cell lines, including viros and CHO cells that don't express B2AR. And when we overexpress B2AR, we then see sufficiency to internalize into these cell types. This is also true in CHO cells. We can also detect an interaction between NS1 and B2AR. We can demonstrate this through co-immunoprecipitation, but here I'm showing you a capture ELISA where we've immobilized ovalbumin, B2AR, or EGFR on the surface of ELISA plates, and we're asking about the capacity of these proteins to capture NS1 in media. And you can see that B2AR more efficiently captures NS1 compared to ovalbumin or EGFR. This is also a functional assay as well, in the sense, in data that I'm not showing you, that NS1 actually acts as a B2AR antagonist. It blocks B2AR canonical signaling. And the reason we think it does that is because we find when we treat HPMEX with NS1, we find a global depletion of B2AR from total cell lysates in a manner that correlates with the maximum internalization of NS1. And this also occurs in mice. If we give mice NS1 intravenously, 48 hours post-administration, we can see a great depletion of B2AR in the blood of mice, suggesting that it may have functional implications. Now, speaking of mice, everything I've shown you so far is in vitro. Now, moving in vivo, I'm going to show you two assays, a vascular leak assay and a lethal challenge assay. This vascular leak assay is what we call BAC-ID, where we shave the backs of mice and administer intradermally different treatments into these spots in the back, including a control like PBS, our beta agonist, isoproterenol, NS1, or NS1 plus isoproterenol. Following this administration, we then give a dextran tracer dye, which is allowed to circulate for 2 hours. We then sacrifice the mice, collect the backs, and then we can measure the amount of leak we have locally in these distinct spots. So as you can appreciate, PBS and isoproterenol do not cause leak on their own right, while NS1 causes significant amount of vascular leak. And just remember, isoproterenol causes depletion of B2AR from the surface of cells. And when we treat with isoproterenol, we can find that this vascular leak is greatly inhibited. What's more is isoproterenol also protects mice from lethal dengue challenge. When we give these mice D220 with isoproterenol, we can find a significant amount of protection, suggesting that blocking this B2AR pathway is important for pathogenesis. So today what I've told you is that dengue NS1 treatment of endothelial cells leads to depletion of protein and then overexpression of the beta-2 adrenergic receptor. B2AR is required and sufficient for internalization and barrier dysfunction of NS1. B2AR interacts directly with NS1, where it also inhibits canonical B2AR signaling, and that pharmacological modulation of B2AR signaling reduces NS1-mediated vascular leak and dengue disease severity in vivo. So with that, I'll conclude. I'll thank my mentor, Dr. Eva Harris, who has given me a lot of room to explore and try new projects, so I really want to thank her for that. And then many different people in the lab who have helped me with this project, including Felix and Laurentia, who are here at the conference today. I'd like to thank collaborators, sources of funding, and you for your attention. I'll take any questions.
Great talk. So I have a quick question. A lot of people take beta blockers. Are there any data from clinical studies where you could look and see people on or off beta blockers? Are they more or less likely to get severe dengue?
Yeah, it's a great point. And some of the clinical collaborators that we've talked with actually state that when a patient enters the clinic and they're on beta blockers, in the time of acute dengue, they usually take them off the beta blockers. What we find in mice is that the beta blockers seem to increase vascular leak. And clinicians have explained to me why that may be the case. And that's why I'm thinking that NS1 may be hijacking this pathway, which is allowing it to cause vascular leak. Conversely, sometimes beta agonists are given to people experiencing severe vascular leak because that also decreases vascular leak. So clinically, there's some precedent for this.
Hi, Lisa Purcell again from Bier. So I guess along those same lines, have you looked for polymorphisms in B2AR, either in people and then that upregulate or downregulate, and then seeing some effects, especially in DHR? I mean, conversely, I think that would probably be pretty tough, but you could probably make some transgenic mice or humanized mice and then introduce those polymorphisms into the experiments in your IFNAR mice.
Yeah, that's a great suggestion. So we've started doing that with collaborators that have those sequences. We haven't made any conclusions yet, but B2AR does have polymorphisms that correlate with asthma, for example, which is a lot about vasoconstriction, which also contributes to vascular leak. So we're hoping to find something like that, but it may be difficult in that sense. But yes, in mice models, we're still hoping to find that, and we're trying to basically map what are the functions of B2AR that are required for NS1. Thank you for your question.